EKG, crustaceans. Therefore, we have been studied crustacean heart as a model of human heart. Crabs' electrocardiograms, EKGs, were analyzed by a random walk analysis technique that we innovated by our group and discovered that dying crab hearts, figure 3, show a low scaling exponent, and healthy crab hearts show a normal SI, near 1.0. Experiments on several animal species, crabs, lobsters, isopod crayfish and insects, revealed that natural death processes decrease SI, falling towards a low level, that is, SI 0.5, figure 4. Then, we encountered strange specimens that exhibited a high SI, such as 1.5. Their hearts stopped suddenly, meaning that they died unpredictably. Figure 5, we noticed that high SI specimens are unique and of rare case. A key observation was that unpredictable death crab always had myocardial injury that was caused by the mounting of artificial EKG electrodes, figure 6. A natural death EKG recorded from a dying coconut crab, Burgess latro. From A to F, decrements in scaling exponents. Immediately after F, the heart stops pumping and fibrillation like electrical signal remained, an arrow. EKG test arena, sea water, and some specimens engaged in the tests. After mounting electrodes, EKGs were continuously recorded for the rest of their life. These specimens were terminally inconvenienced after a period of time, for example, from 2 weeks to 2 years. Unpredictable death. EKG from a crab, Portunist. A similar experiment as shown in figure 3, but this specimen's heart suddenly ceased at an arrow. Note, scaling exponents, SI, are always very high, from A to D. Inside view of a crab carapace. Gazami crab, Portunist. The approximate size of the heart is shown, pentagon-shaped diagram. This picture was taken after the crab's death. Electrode 1, -2, and -3, for EKG, E. Diameter of electrodes, 1 mm. One can see that electrode 3 is too long in size to damage the heart being located immediately beneath the carapace. Myocardial damage caused unpredictable cessation of heartbeat. It took 2 weeks before this crab stopped her heart pumping, which was unpredictable, see figure 5. Figure 5 shows EKG data taken during the unexpected dying process. We normally put 2 EKG electrodes into crustacean dorsal carapace. However, this crab, figure 6, received 3, an excess electrode. As EKG electrodes have no good contact with the surface of heart muscles, they make EKG signal weak. We never want to damage the heart. However, this insufficient condition sometimes occurred. Any electrode can cause this unwished outcome, damaging local myocardial cells. From this unexpected outcome, we accidentally obtained data that proved that myocardial damage increases SI. Then, we had an idea from this crustacean phenomenon that human ischemic myocardium damage might be the same in terms of physiological nature, and damaged human heart might be able to be analyzed with IMPHA, see subsequent text. We believe that crustacean heartbeat continuously persists beating, that is, their hearts beat like the human heart does. But it was not the case, figure 7. With EKGs from freely moving lobsters/crabs, we found that the heartbeat pattern is not continuous but intermittent if animals are not disturbed, figure 7. This intermittency is induced by the activity of cardio-inhibitory nerve, figure 8. Then, EKG analysis revealed that a relaxed lobster exhibits an SI near 1.0 and a nervous lobster exhibits an SI near 0.5, figure 9. These results suggested to us that SI might be useful to quantify the psychology of lobster. Indeed, stressful stimuli decrease lobsters' SI significantly, and electrophysiologically the nervous/stressful state is a state of acceleration dominant and lost inhibition controls of the heart, figure 7. Intermittent stopping manner of heartbeat. Lobster, Ponulirus japonicus. The intermittency ceased when a human approached the lobster tank. Note, an increasing tendency of heart rate during the presence of a human, between arrows. Simultaneous electrophysiological recording, heart, pacemaker, largest spike size, approximately 3 mV, cardio-regulatory nerve, autonomic impulses, largest spike size, approximately 500 µV, and mechanical transducer, myocardial force, the largest peak force of contraction is approximately 1 mg. An increase of a nerve activity corresponds to a complete stop of heartbeat. The smallest spikes in amplitude are the cardio-inhibitory impulses. The other two include the cardio-acceleratory impulses. Hermit crab, Aniculus modified from Izawa and Kuwasawa. A long EKG recording. Lobster, Ponulirus japonicus. A human visit, thick lines, changes the heartbeat pattern. In relaxed and nervous conditions, SI1 and SI0.6, respectively, upper inset. SI distinguishes lobster psychology. We have long been specifically studying the neurobiology of crustaceans. However, the crustacean experiments opened our eyes bigger, and our viewpoint was extended to human hearts. SI measures could be applicable not only to crustaceans but also to humans, at least applying to their time series signal obtained from the heartbeat. According to our guideline, the normal SI ranges approximately 0.8 to 0.90 can be interpreted as skewness and kurtosis parameters, respectively. The parameters are not the direct moments of the distribution. The first four moments, the mean, variance, third central moment, and fourth central moment, respectively, of the distribution according to Yan are as follows. The quantities ohm and omega in the moment formulas are ohm equals gamma, delta and omega equals x delta minus 2. The skewness and kurtosis are jointly determined by the two shape parameters gamma and delta. The standardized Johnson's SU innovations exist when xi equals 0 and lambda equals 1, but the mean and the variance are not 0 and 1, respectively. These can be done by setting the parameters in the following manner.